Okay, we'll move on to our next presenter today. That is Nianwei Lin. Nianwei is the president of iXCells Biotechnologies, and he will talk to us today about streamlining the preclinical solutions for personalized drug development.
Hello, everyone. It's my great pleasure to have a lot of speakers here, including the patient families, sharing the inspiring stories, and also the researchers. We know a lot of exciting progress. So I already a lot of people already mentioned about some facts about rare diseases. I'm not going to go too much details, but it's basically a global crisis. So a lot of our friends and families have been affected around us. That's why I think it's super important to increase the awareness. That's the step one. So, and today, especially for the afternoon section, is we're going to talk about how we can help. So we have a little bit a small fundraiser events here, hosted today and tomorrow and through Thursday. We're encouraging our friends and employees to engage. So if you are here with us today, you're welcome to scan the barcode posted in our door. And the revenue for these 3 days will be donated to our patient families. So I really appreciate everybody's donation. So a little bit background of our company. Some of you already know, and some of you probably never heard of us. So we are a San Diego-based company. We are a preclinical CRO and manufacturing companies. Our specialized, we are specialized in disease-relevant cell models, and especially with the disease-specific models. So we have been growing since 2015, and we start with 5 people, and now we have 40 employees. We are located in the Sorrento Valley areas. So next year we're gonna move to a new facility in close to Scripps Ranch with about 28,000 square feet. Hopefully we can better serve our customers and our rare disease patients. As I mentioned, we are providing the disease-specific cell models. We can help our customer to develop the iPSC cells. We have been working with a lot of recently, especially after 2021. We working with multiple patient families, including there are 2 families already share their stories. We can help them to develop iPSC from human blood. We just need a little bit whole blood, and we can isolate PBMCs. And we can also develop fibroblasts from the patient by skin biopsies. And using the iPS cell lines, we can do genome editing. I'll mention later what's the application using iPSC models for disease modeling, especially for the neural disorders. So I'm not going to go too much details, but we have more than 100 different products. We have more than 70 different primary cells and more than 10 iPSC-derived cells. Some of these can provide as a research toolbox for rare diseases and personalized medicines. I highlighted some of the products and services here. So we're working with a lot of, before rare disease families, we're working with big pharmas, biotech company, for developing very complex neural disorders, neurodegenerative disease, and more recently we have more and more focus and we feel applicated in the rare disease communities. So I presented our company's vision and perspective in 5 years to our board this year. So this is what we're trying to do. Firstly, we are gonna further expand our products and services for sure. And there are 2 new areas we're trying to step into. One is the peptide and oligo synthesis. We are planning to establish our chemistry lab in a new facility. Hopefully we can provide a more cost-effective way to provide a high quality and validated oligonucleotides. And also, we're trying to establish a comprehensive preclinical ASO drug development tools. So a lot of these are we learn from our patient, we learn from our collaborators. I will mention that in my later talk. So as I mentioned, our company, we serve for, we care about our rare patient families. So we've been working with multiple partners. So a lot of these rare disease or individualized drug development is patient-driven effort. We're working with, so these are all some just part of the patient foundation we're working with. Majority of them are here today. I'm very honored to see them to support our events. And we're also working with multiple academia partners and biotech and big farmers, biofarmers. And most of these patients, when they reach to us, they usually have a research committee helping them to understand the patient, the MOA, and they start to design the drug. And iXCells here is really working with them to design the assays and develop the disease models, and yeah, using our expertise in disease modeling. So I want to use a case study showing what we can, how we do the ASO screening. A majority of the patient we're working here has the ion channel mutations. They come with the epilepsy condition. So the patient-derived iPSC neurons is considered as one of the best, most disease-relevant models, although it's more cost-effective and time-consuming. So in this particular case, we're using patient-derived cells, and we culture them for 30 days in 96 well plates. And we treat them in the 96 well plates with antisense, just free uptake method. And then the readout is the multiplex qPCR and using a Pacman-based approach. And the assay was carried out in 384 well plates. And in this particular case, the customer is the patient designed more than 800 antisense oligos, and we screen several rounds, about 7 rounds of screening, and we end up with a few handful candidates, and our partners move further to the downstream tox studies. So this is one of the most challenging cell system we've ever worked with, because the neuron, wherever understand the neuron culture, culturing in the 30 days neuron is not easy. So we have spent almost 3 months to optimize the procedures, and we end up with very clean data here. So you can see 2 identical plates this is basically a QC validation of different some positive control and negative control, and two of the identified hits. We see very, very clean data. So I just want to have another shout out for the data sharing. So this is such a very, very critical steps because individually, all this data is meaningless unless we put everything together. So we want to start, as Winston already shared, that if if the patient is willing to share their data, that's great. We have a lot. Based on my experience interacting with the patient, so there's a lot of challenges when they start working with the academia or biotech. The ownership of the data is it can be a challenging step to share. So here at iXCells, because we are based on our business model, our customer or patient owns their data and IP. So there's no any restriction in terms of the controlling of their data. So at the end, it's gonna be our customers who is signed a contract with us. We're on all the data and IP, and we are ready. I actually talked to Tim and Winston about how to standardize the data deposit and make the data depositing more automatic in an automatic way. And And so those are super critical. But at this time, it's still just a beginning. So iXCells is ready to work with all these nonprofit organizations to streamline the data sharing step. And just a little bit, some of my past years experience with the community. So this is Winston. We have Julia, who is the Mila's mom, and Avalanche founders. And we invite patient family to our facility. It's their first time seeing their their kids' cells. Went to the SLC 681 symposium. I got to learn how these patients become an expert within a very short time frame, and they be able to summon a lot of professionals in different areas. So it's simply just very inspiring. And here, besides the bakery fundraising, we also we're planning to launch our the year-long snack bar fundraiser for our patients. So thanks for my team. They spend a lot of time to put up the poster for each patient. So we also have these posters printed out and hang out in this wall here in our meeting rooms. We're gonna have our have some snack bar, and we'll encourage our patient to learn. Encourage our employee to learn about a story. So to understand, so working with the rare disease here, we're not really dealing with the numbers. We're dealing with the people with the stories. So I hope there are more people will join us to help these families all around the world, or almost 300 million people being affected. So we need to face it, and we need to work with our partners closely. So last part is I want to share some of the experience I learned from the patient and learn from the projects. Hopefully it can give some guidance. I cannot say this is concrete or very crystal for in general, but it giving us some guidance how to build a roadmap, especially for preclinical solutions. As iXCells is really focused on the in vitro models, we cannot give you too much guidance in terms of the understanding of disease MOA, but we can certainly help you to develop a preclinical or in vitro models, and that you can use to accelerate your individualized drug development. So before I mention this my slides, I want to share a roadmap. This is I think this is very useful for a lot of patients who has the newly diagnosed kid. So especially if you don't understand how the drug development process is, this could be a very good guidance for you. You can find a link. This is from Global Gene's website. They provide a detailed explanation of each step from the basic research, what kind of research toolbox you need to provide, including cell lines, iPSCs, mouse models, biomarkers, and what is preclinical, what is clinical, right? And also what is drug repurposing. So I know there's a lot of patients are interested in drug repurposing because this can reduce the long procedure for screening for new leads. And so I highly encourage you to go there, and also they have very fantastic staff that can answer your request with personalized response. Yeah, I don't need to spend too much time here. Everybody here knows what's antisense, why this can be scaled for multiple rare disease. This is one of the modality that a lot of people, including Tim, Winston, and Jeff, everyone, we all think this is probably the most promising modalities to developing the new drug for individuals. So here we can, as I mentioned, we can certainly help the patient to develop their cell models, including fibroblasts, iPSC generation, or making the neurons. We have a dedicated team doing those reprogramming and genome editing in a daily basis. And also this year, we're going to have our partners for making the antisense oligosynthesis. So we're evaluating their quality. Hopefully, we can have some solution for our patients with a much lower price and faster turnaround time very soon. And for sure, we are learning from our past years that we can certainly do the antisense oligo screening with multiple different treatment conditions, different cell lines. And in vitro tox, we have a group of our primary cell groups has a group of scientists are very familiar with isolating immune cells and immune response. So I'll mention that later. So some of those immune response or cytokine profiling assays can be used to predict the toxicity of the compounds. And finally, the in vitro efficacy test is something we are particularly interested. I'll mention that later. So a little bit more detail. So we can, there are several steps to build your preclinical disease models. Firstly is the assay development, including what cell lines to select and what's the treatment, how to optimize your treatment, right? Second step is the hit identification, including the in vitro screening and lead optimization. The third one is the lead validation. So once we have the hits from several hundred ASO to 10 to 20 ASOs, we're down to less than 10 hits. We need to further validate the hits by measuring their, so usually we would do the protein level confirmation. We also, sometimes we need to do the off-target analysis. If we see too much off-target, it can be an indication that your drug is not very specific. So also in vitro disease model, as I mentioned. So this is something I didn't see too much patients doing that, but there are more interest in this space, which is using the 3D brain organoids to study the efficacy. I have a few slides later to show you some interesting findings here. Finally, after you get all these hits and validate your leads, and you can move on to the animal toxicology. So the first step is acute in vivo tolerability. So basically, you are going to test the PK/PD, the dynamics of your compound in animal. And secondly, is you can do PBMC assay to see whether your compounds indeed will cause severe immune response. And finally, you will do more comprehensive clinical, the animal clinical science, behavioral, histopathologies. So because we are familiar with the cell system, so I come up with this table earlier this year to when I talk to some patient, because a lot of patients when they come to us, they are confused, they are overwhelmed with multiple systems. So I list out 4 of the cell systems, commonly used cell system that can be considered for the primary and secondary, and eventually the lead optimization. So I'll start just from left to right. You'll see it's starting from the easiest, from the most difficult. The easiest to make to the most difficult to make. So the patient-derived iPS cell neuron is most challenging ones. So it will take at least 3 months to make iPS cells, another 3 months to make neurons. And also, it's not cheap. You need to get at least 6 months ahead of time, and it can cost $20,000 to $40,000. And also including in terms of the disease relevant, of course, the iPSC patient-derived iPSC neuron is the most relevant to the disease. So it's ideal tool to study to use for hit confirmation and off-target analysis. And certainly, if you want to do in vitro efficacy, that would be great. And from the other side, before you have the patient iPSC neurons, you can start screening using the immortalized cell lines. We have multiple neuroblastoma cell lines we've been used before. I listed it down here. Sorry, this should be a typo. So there's SHSY5Y, which is another neuroblastoma line. So you can use that for primary screening assay development because you need to test whether the gene, you need to design a primer sets and probe, Techman probe sets for your assay. So you don't want to waste time here. So there's also primary rodent neurons and normal iPSC-derived neurons, which is in between of these two, which is not very commonly used. Also, we have tons of questions in terms of the timeline. So based on the timeline estimated for Milasen, I come up with this timeline. As I said, this is not going to be one case for all. So each case is unique. Some case probably will take years to develop. But just use Milasen as an example. So it's giving you a guideline how you design for your assays. So as I mentioned, you don't need to wait for iPSC neurons, which will take 6 months. But I suggest the parents you don't want to wasting your time. Just like Peter mentioned earlier, once you decided to go with the individualized drug development, it's better to have the reagent ready, right? So get your iPSC generated and neuron and iPSC neuron potentially can be useful as well. So, but at the same time, you can also start doing the assay development, talk to your science team, and if they have disease mechanism, if it's gain of function or loss of function, based on your research community, they will suggest you either using the allele-specific or splicing modulator mechanism. We can start doing the assay development in the immortalized cell lines, which is relatively easy to get. We have a list of cell lines we can use. And you can do the hit validation, which includes several dose response assays. So by that time, once you get several hits, hopefully you have the iPSC neuron ready at that time, and you can start using the neuron for hit confirmation or hit optimization or SAR. So basically, once you get the right sequence, you can try different chemistry modification and do different combination of those to see whether you can increase the specificity or efficacy. Another thing is the timeline for ASO design and synthesis. In order to really racing, because we are racing against the clock, we need to plan ahead. Sometimes nowadays there's a lot of delay in delivering the antisense. We are collaborating with several ASO synthesis vendors. Recently, we are facing 3 to 4 months delay in delivering those. So please plan ahead of time. So hopefully iXCells can identify one of our potential partners that can offer faster and cost-effective alternatives. And finally, you can arrange the functional validation and off-target analysis almost at the same time and together with the tox studies. So I have a few more minutes left, and I like to share some of the exciting things I like to share with the community. One of our a new employees who recently joined us. His name is Jorge. He worked in Stemonics. A company was acquired by Vivan Biosciences. They have developed a 3D microbrain organoid approach. So this is just showing you the general process. You can derive the iPS cells from dermal fibroblasts. I believe you can also use the PBMC derived. Make the EB and make it into the neural rosette, further differentiate into the neural stem cells or neural progenitor cells, and you can make a 3D organoids, 3D brain organoids. So here you can see the 3D branch has the markers for both mature neurons and also the glial cells. So here they are trying to get the wild type and Rett syndrome derived organoids and trying to see their functional difference. So this is the assay. Okay, so I'm gonna show, this is on the top is the regular, it's the control organoid showing the rhythmic calcium imaging. So this assay was monitored by Flipper. And down here is the Rett syndrome derived. You can see this is really resembling the epilepsy. So all these peak you are seeing here is the calcium flux, and it will mimic although this is not a real readout, but it's pretty much mimic the neural firings. And the machine can also detect, it can quantify those peaks and magnitudes. And you can see the significant difference between the wild type and Rett syndrome derived organoids. And most importantly, is these organoids can be generated in 384 well plates, and they've done a lot of process development to make sure the size it's relatively equal, and we have very homogeneous size and distribution. Also, besides the flipper, we can potentially use MEA, which is a multi-array, multi-electronic array. So you can, this is the machine. Basically, you're gonna see the cells into different type of plate formats. In bottom of each wells, you have multiple probes which can detect the neural firing. And you'll be able to measure high throughput calcium assays or electric physiological activities. So this is just one of the readout. You see the control. You see this is Raspberry probe. You can see each line is one probe. If you see the purple bar, that means you have the synchronized firing. And for a Rett syndrome derived microbrain, you see a lot of these, very fast firings, irregular firings. If you're firing and stop, which is mimic the epilepsy condition. So finally, I like to I really appreciate my team here. So this picture was taken yesterday for a rare disease for our event today. Our employees, we all wear in the stripes. So with that, I really appreciate everybody's time here. So we're gonna have we will have the recording of our today's meetings. So I know today is not a weekend, so it's a weekday. A lot of people cannot show up. And that's why I really appreciate people spending time with us. We're gonna have the video clips, and we can share with more audience. Hopefully, people can share the words, spread the words, increase the awareness of the rare disease. So as I mentioned, as a lot of people mentioned today, this is collaborative efforts. We cannot conquer this devastating situation by solo.